Good evening, everyone. Today we will start with a few concepts of animal biotechnology. So, first, I will discuss about introduction of biotechnology in brief. Then, we will discuss about more important concepts. So, first, what is biotechnology? So, the term is a new branch, but the application was practiced through many ages. Okay, so it may think like bio means life, technology. So we are applying various processes using living organisms. So that is biotechnology. So this term has appeared recently, like some 10, 20 years back. But so whatever the technology, whatever the processes we are using in under the biotechnology, most of them, the basic concepts were we are practicing since ages, but just a new name was given. Okay, so it is an interdisciplinary. So if you just get the term biotechnology, so within biotechnology you will study many subjects like genomics, recombinant gene techniques, then applied immunology, so immunology, biochemistry, everything we will study, pharmaceutical therapies, diagnostic tests, because it is mostly an application oriented branch of science. Okay, so and it is closely associated with the society because mostly biotechnology means it is innovation of a process or in synthesis of new drugs or proteins or therapies, like that only. So such kind of processes are involved in the biotechnology. So it is more of application oriented. So directly the products go into the market and with the public. Okay, like even some food industries. Okay, then zymotechnology, production of enzymes, and all these things come under biotechnology. Okay, so not only this, so the breeding of plants and animals, then fermentation, so all these were also there since many ages, but now we are doing it with science and technology. Okay, so here we know how to breed the animals. So we know how to get the hybrid plants, hybrid variety of plants. And the ultimate goal of all these experiments is to get some useful product out of it, which is for human application. Is it? Is it clear? So the major areas are healthcare, food technology, and environmental protection. So these are the major concepts. Okay, just a second, I will just continue. Yeah, sorry. So, so the applications of this biotechnology are majorly used in food industry. Now we know like new food varieties using microorganisms. Okay, and we are what you call we are enriching the nutrients within the food. So already the food are available. So we are actually enriching, like if you see golden rice. So the rice variety is there, but we are inducing new genes so that its nutritive value is increased. Okay. Then flavor saver tomato, which increases the shelf life of the tomatoes. Okay. Like that, we will use this biotechnology to increase the nutritive value of the food and to preserve food, then to prevent human illnesses. So various vaccines or uh, insulin. So insulin was the first initial products which was developed using biotechnology. So insulin, such kind of proteins or therapeutic agents were also developed. Then we have environmental biotechnology where we clean up the water or waste, etc. Okay, so these all comes under this biotechnology. And the most important processes are recombinant DNA technology, then cell fusion, how to fuse, then bioprocess, that is to separate or purify some products, then structured based molecular designing. Okay, so I will just, these are just, I am discussing briefly. So I will give one example like insulin, how it is produced. So previously, insulin, so the diabetes used to come to only few people. That's why it used to be called as rich man's disease. So only rich people used to get, and they only used to get the treatment also. But nowadays, almost everyone, 90% of the people are having diabetes. Okay, so for this, so in previous days, they used to kill the pigs, they used to isolate pancreas, they used to isolate insulin from pigs, and then they used to inject that insulin. But that you cannot do it now because even ethical laws are very strict, as well as how many pigs will you kill, right? So the percentage of people with this disease has increased. So people should look for alternatives. So for this process, they have isolated insulin gene, and then they have introduced this insulin gene into a vector. So this vector is usually plasmid, but many other vectors like cosmid are also being used. Okay, then that was introduced in the E. coli, which is bacteria. So bacteria E. coli has a generation time of 20 minutes. So that is every 20 minutes it replicates. So one E. coli will become two, two become four, like that. So therefore, the gene is also replicating very quickly. So once it is replicated, then this gene has to be introduced into the expression vector where the gene gets expressed. Once it gets expressed, then it should be purified. Okay, just hold a second. There is some technical problem. I will just. Yeah, sorry. So this is how. So once the purification is done, so then that protein can be marketed. So this is one kind of mechanism that is called as recombinant DNA technology. So in the same way, many proteins can be synthesized and can be expressed. Along with this, yesterday I told you hybridoma technology. Even that is a product of biotechnology. Then bioprocess technology. So which will help in, so bioprocessing means we are doing various processes within a plant or animal. So bio means life. So within an animal or plant, we are exploiting some process to get a useful product out of it. Okay. So I'm not discussing in depth because you have another important topics. So I will just briefly explain. You can go through. If you have any doubts, you can just get back to me. So now there are various branches within this biotechnology, that is medicinal biotechnology, industrial biotechnology, that is environmental biotechnology, then agriculture biotechnology, like that. So medicinal biotechnology is mostly related to drugs, okay, genetic gene therapy or genetic engineering, all this comes under medicinal biotechnology. So it is a gene therapy. Gene therapy means replacing the defective gene with a healthy gene. That is called gene therapy. Okay, like that, various dysfunctional genes were already the gene therapy got approval, and so in some other countries this is already implemented, but not in India also in some cases. Okay, then industrial biotechnology is the production of various drugs and chemicals like lactic acid, glycerin, etc., and some enzymes, immobilized enzymes. All these were produced, which are having economic importance. Then environmental biotechnology. So here mainly to clean up the environment, to clean up the water, air, as well as soil. Okay. So best example is superbug, production of superbug. So here usually this oil tankers will be transported through ships. So if there is any accident to the ship, then all complete oil will be spill over the water. So as you know, it cannot be mixed with the water, it cannot dissolve in the water. So it will form a layer. It is not just 1 liter or 2 liter, right? Because it is a tanker, so some thousands of liters of oil, it will form a layer above the water. In such cases, what happens? You have to clean up. So only way is evaporation. So they used to um, light up, okay, just they used to fire it so that complete oil get exhausted and it will be cleared, water will be cleared. But the disadvantage is that it produces lot of heat. So when there is heat, all the organisms, aquatic organisms present, they will die. Okay, their existence will be questionable. Okay, so what biotechnology has um, given is that the scientists have developed a bacteria which is called as superbug. So it can eat oil. So they used to spill this bug over the sea, and it used to clear the complete, it used to eat complete oil layer on the sea or ocean. Okay, so this is one of the what you call technology which was developed using biotechnology to clean
Ma'am,
up the, sorry,
it is which bug?
it is called superbug.
Super bug.
Yeah. Okay,
Okay, ma'am.
then various biopesticides, so such kind of many products were developed. Then agricultural biotechnology. So now we know like pesticides, most of the pesticides are developed. So when pesticides are developed, insecticides, they will their residues remain intact in fruits and vegetables, and again when we consume them, they will be transmitted to our body. So now that's why people are saying you have to wash with water, salt water, hot water, like that we have to clean fruits, vegetables 2 or 3 times, and then only we have to consume. Even studies suggest that even in the breast milk of mother also pesticides were detected. So that means since newborn stage only people are getting exposed to various pesticides. So although it improves the production of crops, but again it is causing various diseases like cancers, etc. So for this case, usually this bt cotton, bt crops, so by genetically modified crops, so they were developed. All these technologies, the basis is mainly for sorry. Yeah, so all these are a part of biotechnology innovations. Then you have different terminologies like red biotechnology, green biotechnology, blue, white, non-gene, gene biotechnology. So red biotechnology involves employing the stem cells and other tissues. Okay, so they are for the production of novel drugs or to regenerate organs. So nowadays organs are also regenerated from the stem cells. So that's why stem cell banking also has become popular nowadays. So before birth only, like just during birth, they will cut this umbilical cord, and this umbilical cord stem cells will be preserved. Okay, so that in future, if there is any disease for the baby, so these stem cells can be used. Okay, then this all comes under medical biotechnology. So that is called as red biotechnology. Then green biotechnology. So it is mainly for agriculture purpose. Okay, for production of pest resistant grains and disease resistant animals. Then blue biotechnology. It is for marine and aquatic environments. Okay. Then white biotechnology. It is mainly regarding production of new chemicals and new fuels, like we know biodiesel, biopetrol, all these, because the fuels are also now fossil fuels, they are exhausting. Like if we use in this space, so it in next 100 years complete reserves of the fossil fuels will be depleted. So new alternative sources of energy has to be invented, or they have to develop. So in such cases, this white biotechnology came into picture. Now non-gene biotechnology. So here there is nothing to do with the genetic level. So complete plant tissue culture at tissue level, or seed production, fermentation, hybridoma technology, then immunochemistry. All these are at protein level or tissue level. There is nothing to do with the genetic level. So gene biotechnology is the cloning. That is, we are transferring the gene, or gene therapy, genetic engineering. All this comes under gene biotechnology. Is it clear?
Yes, ma'am.
So now we will discuss about what are the important molecular techniques in gene manipulation. So this topic is important. Okay. So here we have cloning. So biotechnology is mostly involves gene manipulation. So first thing is we should identify the organism. So gene of our interest. So I will take example of insulin again because it is well known one. So I want to produce insulin protein. So first is I have to identify insulin gene. So already insulin gene was characterized, its sequence was characterized, its chromosome, its location, everything was identified. So it is not much difficult. And I want human insulin gene. So first I have to take that particular gene. I have to isolate the DNA. I have to isolate that gene. So how to amplify that gene means I'm not taking only single copy of gene. I need multiple copies of the gene. I have to amplify. So for this, polymerase chain reaction, PCR,
Ma'am,
is a technique. Yeah, sorry.
amplify means?
To increase the amount. Okay, that means, see, another example I'll tell where PCR is actually used. So in the forensic science, suppose there is a crime. Okay, so you can't find so much of samples. See, for any experimentation, we need some a minimum amount of sample, right? But in crimes, suppose you are taking some blood test, normal blood test from a patient, you will take 5 ml of blood sample so that you can perform the test. But in a crime scene, you cannot expect 5 ml of the blood, or you cannot expect enough amount of sample from the um, convict who has done the crime, right? So what to increase, but you have to do the test. For doing the test, at least you need some 10 picogram, but you got only 1 picogram. So what to do? You have to perform the test, no? So in for such cases, PCR, polymerase chain reaction, was developed. So here, do you have idea about this technique, PCR?
No, ma'am.
No, ma'am.
Okay, 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 fine. So for this case, actually what they have done is So we have to take the DNA. Okay, DNA, and there will be primers. So primers are nothing but small another complementary sequence. So you know the DNA sequence, right? So what is DNA? It will have two strands. All these
Yes,
basic
ma'am.
concepts. Sorry.
Yes, ma'am, we know.
Okay. So DNA usually have two strands. So one is complementary to the other. Now what we have to do is, suppose A, T, G, C, 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 T something is the sequence. So first we have to sequence the DNA. Okay. You have sequencers. So just add the sample, you will get the sequence. According to the sequence, you have to design the primers. Primers are nothing but short, some 10 to 20 base pairs, short stretch of another nucleotides which are complementary to the strand which you want to increase or amplify. Okay, that is, I am giving you example. Suppose A, T, G, C, 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 something is there. So A, what is a complementary to A? It is T. For T, it is A. Like that, you have to synthesize a small stretch of nucleotide, which is called primer. Once you add the primer, you take small tube, you will add DNA sample, you will add forward primer, reverse primer. Two primers will be there. Then you have to add them. And to that, you will have a buffer. Then you will have tag DNA polymerase. So polymerase is an enzyme which will help in the amplification. Okay, polymerization. So that you have to add. And all these once you add, and DNTPs, deoxyribonucleotide triphosphates, for the synthesis of new strand. So all these you have to add. You have to insert that in the PCR machine. You will have a program. So in the PCR you have three steps. First is denaturation. Now the two strands, DNA strands present in the sample, they get separated. Then second step is annealing. Annealing is nothing but the primers goes and binds to these two strands. And third one is the like polymerization. So they will start adding DNTPs according to the template strand. And this continues for around 30 plus cycles. So usually during program, you can keep it as 30 cycles, 40 cycles, 50 cycles, like that. You have to optimize it. You have to standardize it. So after the program, you will get, so you have to just, after one cycle, one DNA strand will become two. In the second cycle, two will become four. Okay, like that. According to the number of cycles, the amount of DNA will increase. Now you have enough amount of sample to perform any test further. Okay, is it clear?
Yes, ma'am.
You can go through, you have videos, YouTube videos and all that. Once you go through for the concept. Okay, so this is called PCR. Now we use this PCR and all these things during our gene manipulation technique. Okay, so first step is isolation of DNA. First you have to identify the gene, what you want to do, which gene you want to express. Suppose insulin, you want to express the insulin protein. First you have to isolate the DNA. Then this DNA in the sense not complete genome. Okay, only that gene. That gene you have to insert in the vector. So what is a vector? It is a plasmid usually, that is extra chromosomal DNA present in the bacteria. So bacteria have circular DNA. Along with the DNA, it will also have some other circular DNAs which are called plasmids. They are extra chromosomal DNA. They are not required for bacterial life. That means for survival of the bacteria. So what scientists have done is they have isolated these plasmids. They used them as vectors. So they will cut this plasmid at particular sites using restriction enzymes. Okay, restriction enzymes are the enzymes which will cut the DNA at specific sites. And the sites are called as restriction sites. Okay. So already scientists have characterized like which site, which enzyme it cuts where. So you also have various restrictions enzymes in the market. You can directly place the order. You will get the restriction enzyme along with where it cuts and all these things. Okay. All the research has almost done. Okay. Only thing is you have to apply that wherever you want. Now, once it is cut, when the vector is cut, then you will insert this gene in that place. Now you have a vector. So I'll just show you like this. So here you can see this green color one is the vector and this red color one will be the gene of interest. Like this, it will be cut and it will be, now this is the recombinant vector. Okay, recombinant DNA. So this will be again inserted in the same bacterium. Like if it is removed from the E. coli, again they'll place it in the, yeah, they'll place it in the E. coli. Okay. So it replicates along with the E. coli. So you will get multiple copies within no time. Okay. So once you get the copies, number of copies of this gene, then you will have another vectors which are called expression vectors. So do you know replication, transcription, translation?
Yes, ma'am.
Okay. So any gene to get expressed, it requires some machinery, right? Some enzymes and some promoter region, terminator region. All these things are required. RNA polymerase, then ribosomes, all these are required for RNA synthesis and protein synthesis. So all this will be available in the expression vector. Okay. Hence it is called expression vector. However, this is called as cloning vector only for cloning purpose. So then once it is expressed, then it will be sent to the fermenters. So where the downstream process, this protein will be filtered, it is purified, and then it is released into the market. Because directly the protein cannot be, because there will be some impurities. So because within the cell, along with the protein, you will also have DNA, RNA, other enzymes, all these things. Then cell debris, all this. So it has to be purified before marketing. This is the overall uh, thing in the gene cloning or gene manipulation. So now here you can see gene cloning vectors. Okay. Sorry, first before that, you have cutting of DNA is done by endonucleases, restriction endonucleases or restriction enzymes. So they cut the DNA at specific sites, and again joining is done by DNA ligase. Okay. Then this cloned DNA should go to DNA sequencing, then expression of cloned genes, then purification of cloned genes, and their localization, their activity, everything should be tested. So first is cloning vectors. So cloning vectors are the autonomously replicating genetic elements or extra chromosomal genetic elements where they carry this genomic DNA into the host cell for gene cloning. So first it has to cut with the restriction enzyme. So they should have restriction sites. Okay. Once they are cut, so another important concept is it should not interfere with the gene of interest. Okay. And the vector should consist of restriction enzyme cleavage sites because the restriction enzyme has to cleave. And they should also have suitable marker. So see, we don't know, like we cannot see with our naked eye whether the plasmid has entered into the bacteria or not. We will do it as a protocol. But after some generations, we should check whether how many bacteria are actually having this recombinant DNA. Okay, for that we will use the media for selection of those bacteria. For selection, usually suitable marker will be there. Like for example, if it is having ampicillin resistant gene, that is, if the plasmid is having ampicillin resistant gene, that bacteria is resistant to ampicillin. Ampicillin is an antibiotic, right? Suppose if this vector is there in the bacteria, it can even grow in the media having ampicillin resistant ampicillin. Now what we will do, like after some generations, we will prepare a media with ampicillin and we will grow that bacteria. Now only bacteria with this recombinant vector will grow because it is having resistant gene. But other bacteria which is not having this gene, they will die because they are sensitive to ampicillin and they will die. So this is selection process. Such kind of many selection processes are there. Okay. And then it should be easy to carry this vector into the host. So we have different vectors: plasmids, bacteriophages, cosmids, phagemids, artificial chromosomes. So what are plasmids? They are present in the bacterial cell, extra chromosomal DNA, double-stranded DNA. They can replicate on their own. So it depends on the same enzyme for replication of the genomic DNA. And with every cell division, this also will replicate. Okay. And it will have a resistant gene for antibiotic, and it will be having restriction sites. Okay. So all these makes it suitable for using it as a vector. So it can be of single copy, that means only one plasmid per cell, or multi-copy. So its copy number, that is called as copy number, 10 to 12 plasmids per cell. Or plasmids under relaxed replication control can also have up to 100 in number. And it should be easily isolated from the host cell. It should have only single restriction sites or one or more, means single restriction site which can be cleaved by one or more restriction enzymes. Then it should have the capability to insert foreign DNA and help in the replication, and it should be reintroduced back into the host bacterial cell. Okay, so mostly we have E. coli is mostly used. Okay, so here, so here there is a another thing called multiple cloning sites. So here, what is multiple cloning sites means? So this vectors are modified to increase the copy number in the host cell. That is by the process called amplification. And these vectors can be constructed by a polylinker or multiple cloning sites. That is, you are increasing the multiple copies or multiple linkers to join together so that you can have multiple cloning sites within the same plasmid, so that it will increase the chance of amplification of the gene of interest. So PUC 19, then PBR 322, then PBR 318, PBR 320. All these are the different plasmids which were actually constructed also. Some of them are constructed. Okay. By taking different pieces from different vectors. Okay. So these are the examples. So here you can see PBR 322, you have ORI, that is origin of replication, from where the replication starts. Then TET, that is tetracycline resistant gene. AMP, that is ampicillin resistant gene. So here these are the restriction enzymes: PST1, EcoR1, HIM3, EcoR4, BAMH1, SAL1, NDL1, NDE1, all these. So these are the sites where actually you can cut and you can insert the DNA. Okay. So these are plasmids. Then bacteriophages. So bacteriophages are the viruses which infect bacteria. So even these bacteriophages can be used as vector so that it can introduce this foreign DNA into the bacteria. So if the insert or genetic material is more than 2 kb length, then bacteriophage is used. Okay. Okay. If it is very small, plasmids will be used. So phage lambda. So that is the commonly used one, lambda phage. Okay. So it will have lytic and lysogenic cycles. So viruses will have lytic and lysogenic cycles. So lytic cycle is nothing but this viral DNA, it replicates bidirectional in the two directions by using a rolling circle mechanism, and new virions, new DNA molecules are synthesized. Okay. And again, it will form protein coat and it will form the shape of the virus, and then it will cleave the host cell and the virion comes out as a new babies. Okay. So it is a destructive phase. In the lysogenic, actually, they won't kill the host, but all the processes, the mechanism, this is the pathway, replication of lambda phage. So these lambda genes will get replicated by using the machinery of the host cell. Okay. And they will live along with the host. So then here the in vitro packing takes place, everything, protein coat will be formed. All these are important. So all the important packing, everything will be done, and they will be slowly released, but not completely cleaving the bacterial cell. So here the lambda genome consists of some genes which are required for the lysogenic cycle but not for the lytic function. Therefore, these genes are removed and they will be replaced by the foreign DNA up to 23 kilobase pairs in the length. Okay. Then they will be introduced. So when this virus infects a bacterial cell, this genomic DNA will be introduced back into the bacterial cell. So here the bacteriophage vectors are EMBL3 and EMBL4. Okay. Now this non-recombinant DNA resulting from the fusion of left and right arms will give the DNA which is small. Okay. And they will be selected for the recombinant phages. That is, okay, once you have introduced this recombinant DNA, again here it is not 100% that every bacteriophage will get the recombinant DNA. So again the selection process has to be done. So two vectors have polylinker with reverse orders of restriction sites with respect to each other. So that is called insertion lambda vectors. So where the vectors are used for making cDNA library. So what is cDNA? Which is complementary DNA. So usually from DNA, RNA will be synthesized. But here in RNA viruses, from RNA, DNA will be synthesized. So they are called as cDNA. So cDNA libraries will be produced for sequencing or for genome sequencing projects like human genome, all these things. So usually in the viral genomic products, this cDNA libraries will be developed. So in that, actually this lambda vectors will be used. So the vectors used here are lambda GT10. Okay. And it allows the screening of recombinant phages. So once it enters the E. coli, then actually this has to form plaques. Okay, plaque is nothing but like a colony after it leaves the bacteria. Okay. Suppose this E. coli strain is having this mutation, HFLA1 150 mutation. Okay. So that is mutation in the lysolytic phase. Sorry, lysogenic phase. Okay, that means this lysogenic pathway is defective and only Cl phase is there. So it will form Cl plus will form lysogens and will not form the lytic cycle to form any plaques. Okay, so this is how the like selection process. This is a type of selection process. So no need to remember that much depth. Just remember the characteristics of the these vectors. Like first is plasmids. So what are the plasmids and all these things? So what are the characteristics of plasmids? So why it is used? Then examples. Then bacteriophages. So what are bacteriophages? How they can be used as vectors? So you can briefly explain about lytic and lysogenic cycle. So you have the pathway right here. Here that pathway you can show lytic and lysogenic cycle both are there. Then you can just give the examples of these viral, that is bacteriophage plasmids or vectors. So then next are cosmids. So in the cosmids, so they are formed by inserting cos sequence of the phase. It is nothing but a hybrid. So cos sequence of the phage into a small plasmid vector. Okay, here you can see cos site, then polylinker where you can insert, then ampicillin resistance, which is a marker. Okay, E. coli 1, that is origin of replication, ORI. So all this will be mixed up to form a new vector. This is called as cosmid vector. Then phagemid vectors. So they are also artificially combining bacteriophages with plasmids. So here you will have, just a second. Yeah, this is phagemid. So this is the selection marker, green color one. Okay. Then this is the replication of origin for plasmid, origin of replication. Then you have promoter, then signal peptide, restriction enzyme sites, then coat protein and Ig region for production. This is a phage. Okay. So for this is called helper phage. Okay. So these are the genes from bacteriophages. Then here you have yak vector where artificial chromosome is also developed. So here you have E. coli R1, then origin of replication. E. coli R1 is the restriction enzyme. Then marker A, marker B, origin of replication, like that. Okay, ampicillin resistance. All these, they will be mixed up and to construct a new one. So these are the yeast chromosomes. So sen, tel, all these things. Okay. So artificial chromosomes, like bacterial artificial chromosome, bacteriophage, P1 or P1 derived artificial chromosome, and yeast artificial chromosome. So, bacterial artificial chromosome is nothing but where the cloning DNA in bacterial cells have high to moderate number of replicas. So it is highly stable. It will have highly stable inserts. So here large DNA can be inserted in the bacteria and they have low copy number of replicons. Okay. And a factor called fertility factor or F factor from E. coli was prepared. And here two genes, par A and par B, are there to maintain the copy number of the F factor. So at least 1 to 2 copies of F factor will be present in the each E. coli. and the DNA vector can be transferred into the bacterial cell. So it's an artificial chromosome. It was constructed by taking these genes and segments from different vectors. Then bacteriophage P1 vectors are packed vectors. So bacteriophages have relatively large genomes compared to plasmids. So here some aspects of plasmids and some aspects of bacteriophages like protein coat and the cloning site, all these are from the bacteriophages. Okay, but the origin of replication, then markers, these are all from the plasmid. Okay, like that they have clumped. So here up to 100 kb of the foreign DNA can be ligate. Then yeast artificial chromosomes. So very large DNA fragments can be cloned using yeast artificial chromosome. So a yeast telomere genes, then centromere sequence, then a marker which is TRP1 or URA3 for detecting the clones. Okay. Then autosomally replicating sequence, that is called ARS or origin of replication. All these are mixed with the yeast and bacteria. Then that is marker genes and all that. Now the resulting yak cannot be transfected directly into the yeast cell because they have external cell wall. So they should be treated, and the resulting yeast spiroblast can accept these fragments, and now they can be inserted into the yeast. So these are the artificial vectors which are commonly used. So here for this purpose, you just remember different vectors. So remember these points. So what are the different types of cloning vectors? So what are their properties like plasmids, bacteriophages, then artificial chromosomes, all these artificial vectors like BAC, YAC, all these. So what are its advantages? Like how much amount of DNA can be inserted? So what are its applications? You should remember the figure. So first remember the figure. So what are all the components of the figure? Then you can explain that figure. So it will usually have a marker gene, origin of replication, then you will have some genes from the yeast. Okay, like that, the combination. Is it clear? This is how you have to answer this question if it comes. Then cloning methods are cell cloning. So cloning of a single celled organisms like bacteria usually. Okay. So here single-celled organisms like bacteria or yeast can adopt this process of inoculation. And a tissue culture technique is used to distinguish cell lineages in the usage of cloning. So here, what is the purpose of this cloning is to produce some drugs, okay, or for testing. Then stem cell cloning is also done. So here the somatic cell nuclear transfer in stem cells. That is, somatic cells are the vegetative cells, non-reproductive cells. So the DNA or nucleus from the somatic cell will be isolated, and that is suppose normal skin cell. So from that skin cell or mammary gland cell, so the DNA or nucleus will be isolated, and that is inserted into the egg cell. Okay. So now, after fusion, you will get the organism which is having the genetic material of the somatic cell. So using this way, a sheep called Dolly was actually developed. Okay. So Dolly was the first mammal which was successfully cloned from the adult somatic cell. So from the udder, that is mammary gland of the female cattle, sheep, goats, okay, and related biological mother, and that was introduced into the egg, denucleated eggs. That means the nucleus in the egg was removed, and this nucleus was introduced there. Okay. So from the biological mother only it was taken, it was inserted into the sheep ovum. Okay. After 434 attempts, actually, it was successfully done. Okay. So, a female sheep was born through normal pregnancy. This was done by British scientists in the Scotland, Rosedale Institute. And the sheep was named as Dolly. Okay. Sir Ian Wilmot and Kate Chamble. Okay. So they have actually developed this dolly in 1996, but it was died after 6 years. So this stuffed animal was already placed in the Royal Museum, even in the nowadays also, like you can see that in the Scotland. So here this is the process how the cloning was done. So Scottish blackface, it is the cytoplasmic donor. So this is the nuclear donor, Finn Dorset. So mammary glands were taken, and here egg was taken. So from egg, nucleus was removed, and from the mammary cells, the nucleus was removed and inserted into the egg. And from after fertilization, in the it was placed in the surrogate sheep, and the dolly was born. Okay, this is clone. So this is how we can actually do cloning to improve milk production in case of cows, to improve woolen production in case of sheep, like that, in meat production in some animals, like that, which we can implement this research in real time, industrial scale. So here, this gene cloning is nothing but a process of um, joining the fragment of complete genes. So now we have to first sequence, then amplify, then we have to insert, and we can use this for the biological experimentation and practical applications. So here in this process, the DNA fragment involves 4 steps. First is fragmentation. It has to be broken apart. It has to be separated. Then ligation. This DNA the gene of interest should get ligated to the vector. Then transfection. It has to be introduced into the host cell. Once it is introduced, we have to screen the cell. Once screening is done, then we can isolate the product out of it. Either it expresses some protein for production of a phenotype like wool or milk production or so and so, or we can actually we can isolate and purify the proteins of our interest. So these are the applications of the gene cloning. Okay, in the next class we will continue with the animal cell culture. So is it clear with the today's class?
Yes, ma'am.
Okay, just go through once, because discussing, like, it is very lengthy topic, so I'm just explaining in your brief. You go through once, you read once, you can get lot of resources from the YouTube and even in the net, like if you want the basics of the principles of the techniques. Okay, you can visualization will be more, even about dolly. So what are the other innovations that have come? So even flavor saver tomato, bt crops, all this, that information you can find. So that will be easy for you to understand rather than simply reading. Okay.
Yes, ma'am.
Okay, fine,
Okay.
thank you so much. Yeah.
Thank you. Thank you, ma'am.